we'll discuss an important topic in biotechnology, that is gene editing. Now first of all, we look into what exactly is this gene editing and the process that is involved in gene editing. Now as the term suggests, gene editing means you edit the genes. Now what do you mean by this term gene? Now gene can be explained as a segment, in simple terms you can explain it as a segment of the DNA, and DNA stands for deoxyribonucleic acid. So gene is a segment of the DNA and that contains the information for the synthesis of a protein. Now the synthesis of that particular protein means either the presence or the absence of any particular trait. So that is what is the importance of genes. Now gene editing means it is editing the genes, and it is a type of genetic engineering where the DNA is inserted, deleted or replaced in the genome of an organism to treat a particular disease. You can also explain it as the synthetic modification through molecular scissors. So we will understand in detail. So this is the overview of what exactly do you mean by the term gene editing. Now why exactly this gene editing was in use is because of two reasons. First thing is that last year China did a successful experiment using this technique of gene editing where it made use of this CRISPR Cas9 technique to inject a gene in a patient with a lung cancer. And recently this year United States went a step ahead and it edited the genes in a human embryo using the CRISPR Cas9 technique. Now why exactly did they go for this editing of the genes is to correct a genetic mutation that thickens the heart muscle, and that condition is called as hypertrophic cardiomyopathy. So hypertrophic cardiomyopathy is a disease that is caused by the thickening of the heart muscles, and it is said to affect about 1 in 500 people and cause sudden death in the otherwise healthy athletes, and it is mainly caused by the mutation in a particular gene, and that gene was mainly edited through this CRISPR Cas9 technique. So we look into what exactly is this CRISPR Cas9 technique. Now the CRISPR term stands for clustered regularly interspaced short palindromic repeats, and it is a system that is mainly found in the bacterial cells to recognize and destroy the viral DNA as a form of the adaptive immunity. So it is mainly to identify as well as destroy a viral DNA. Now the system mainly consists of two components. The first is the single guide RNA, or what you call it as the sgRNA, that contains a sequence that can bind to the DNA. Now what is the specialty of this particular RNA is that it contains a sequence that will be matching the area or the part of the DNA that is to be edited. Or else you can say that the single guide RNA helps in targeting the area which is to be edited in the DNA. So once it targets, this Cas9 enzyme gets activated, or when it reaches a particular target, this Cas9 enzyme gets activated and it acts as a molecular scissors that can cleave or edit that particular DNA. So this is how the system works. So you can say that the single guide RNA guides or help in bringing this Cas9 to the target area, and the Cas9 system is the one that cuts the DNA and make the according changes. So once this DNA is being cut, you have this DNA breaking points, and that will be mainly corrected by the inbuilt DNA repairing system that is present in the body. So this is how this CRISPR Cas9 technique mainly works. Or else you can say that you have this particular DNA nucleus, and you have the DNA, and this guide DNA attaching it to the area which is to be edited, and this DNA cutting enzyme will cut that particular area, and if it is to replace, the healthy DNA strand will be used to replace that particular DNA. So this is how the CRISPR Cas9 system mainly works. So looking on to the potentials of this particular technique, the first potential is in regard to permanently fixing or editing the mutated cells. That is, the gene editing provides a one-time treatment for the patients who are suffering from a broad range of diseases. Now suppose the person who is suffering from HIV, that person can make use of this gene editing technique. Now I've already mentioned about the experiment that was recently conducted, that is the human embryo was being edited for a heart disease. Now the advantage that has been cited by the scientists is that the resulting babies will not only be free of this heart disease, but also this disorder will not be passed on to the future generations. So that is another advantage in regard to the gene editing. So the first is permanently fixing or editing the mutated cells. So that is the first thing. Second thing is that it can be also used to prevent the diseases that are partly genetic. So what are the examples? You can say the diabetes as well as cancers that are to certain extent genetic, So those can be, that particular gene can be edited and this disease can be prevented. And next is its usefulness in confirming the functions of various genes. For example, you have in Japan the gene editing experiment was being conducted with a particular gene, and that has resulted in the change of the color of the flower from violet to white. So they were able to understand that this gene stands for this color. So in that way, through gene editing, we can confirm the functions of the various genes. And next, it can be also used in agriculture, that is to improve the crops. And then it can also be used to make the organ transplants more efficient. That is, recently the gene edited piglets were being developed, and these were free of this retrovirus, which was considered to be causing diseases to the humans. So these can be various potentials that arise out of this gene editing mechanism. The first thing is that it permanently fixes or edited the mutated cells, and second, it helps in preventing the diseases that are partly genetic, and it is useful in confirming the functions of various genes, and it is used in agriculture and also in organ transplants. But there have been concerns in regard to the gene editing. Now what all are the concerns? So the first concern is in regard to creation of designer babies. Now what do you mean by this designer babies? Now these are the babies who are having the desired characteristics, and that has been brought about by altering the embryos. So that is the first concern, that is creation of designer babies. Second is in regard to the safety concerns. Now when you consider this CRISPR Cas9 system, it is one of the most precise gene editing tools present. But the problem is that you can never ever remove the possibility that this particular system can recognize as well as edit a particular segment which was not the intended segment. So such kind of off-target chances can never be eliminated. So that is another concern, safety concerns. Third is in regard to the problem of accessibility. You have the genetic techniques most often being very expensive, and if this gene editing tool is not going to be made available or accessible to the most of the population and just remaining to only the rich segment, that will be very problematic. So that is the problem of accessibility of the treatment. And then next is in regard to the chances of the misuse. Now we have examples where the surrogacy was being misused. So similarly, you have this gene editing comparatively considered to be simple, and there has been the initiatives by various communities even to train the lay people about this particular treatment. So what happens is that there are chances that this can be misused if proper regulations are not placed. So there is always chances of misuse. Another concern that has been raised is in regard to use of this gene editing technique as weapons of mass destruction. That is, if it falls in the wrong hands, it can be used to edit certain genes, and that can spread the spread virus or even affect the food chain. So such kind of possibilities are also being raised. So these are the main concerns in regard to the use of the gene editing technique. The first is creation of the designer babies, and second the safety concerns, third the problem of accessibility, fourth the chances of the misuse, and fifth is in regard to the potential weapons of the mass destruction. So what is the way forward? First of all, we need to ensure that there is in place a proper regulation, that is to look into how or who can make use of this gene editing and how, under what circumstances it will be used. So that kind of regulations should be in place, and for that the international community has to come together. Now coming to India as well as this gene editing, India is not in the forefront when it comes to the gene editing technique, but India can take the initiative in starting the discussions or international discussions about the access to the gene editing technology. So that is one thing that India can look into. And second thing is that it should develop domestically or within India, it needs to develop the capabilities in this gene editing field and consider how it can benefit the Indians medically as well as in an inclusive manner. And for that the government needs to allocate more resources, and these need to be ensured. So this is in regard to the gene editing, its analysis, its pros and cons,